Well, hi, everybody. Welcome to the Innovations in Neuro-Oncology. We really appreciate you taking the time to let us tell you about some of the stuff that we're doing, and maybe we can chat in sort of a different way that we might from the clinic. So we're really grateful that you're here, and we're going to just talk about some of the things that we're working on in general. There will be questions after that. I think you can submit during the chat. So there's going to be 3 of us presenting, and then we'll open it up for questions. Rael, do you do the next slide? There you go. So I am obliged to read this. The content is not intended to be medical advice. The viewer should consult their physician should they have any questions. The viewer should not rely on information contained in this presentation or webinar for immediate or urgent medical needs. Additionally, if you think you have a medical emergency, call your physician. Go to the nearest emergency department or call 911 immediately. Never disregard professional medical advice or delay seeking care because of information contained in this presentation or webinar. So I think one of the main points that we really wanted to get across is just the kind of size of the team that we have that winds up looking after people with these complex problems, particularly for tonight, talking about brain metastasis and leptomeningeal disease. These are all problems that occur from cancer in the rest of the body. So it's really complicated because we have to deal with cancers in the rest of the body as well as what happens when they go to the brain. So it requires a very large team. So we're lucky just in the medical side to have 6, soon to be 7 medical attendings, such as myself, Dr. Grogan, Jaffer, Macari, Figueroa, and Pina, as well as a number of APPs or physician's assistants, which are several, all of which are fantastic. And then in terms of training, we have a couple of fellows, Dr. Elza Farani and Dr. Boldig. And then neurosurgery is part of my department, which there's 5 neurosurgeons. Dr. Vogelbaum is the chief. And they really do a fantastic job. They're very dedicated and very patient-oriented, I would say, and they're great colleagues, as well as a number of APPs or physician assistants that really help us do all our work. And then we are a institution of higher learning, so we do have fellows in neurosurgery as well. And of course, all our fellowship programs and educational things are all certified. Do the next one. And then you may have seen many of the people that I just showed you, but we're also really just part of a bigger effort. So we in turn need a lot of support. A lot of our work has to do with reading MRI scans or every once in a while a CAT scan. And for that, we have this wonderful team in neuroradiology that helps us read our MRI scans. Some of you may have noticed, but we've asked them and they've agreed to actually sit in a room just off the clinic space. So if someone has a problem or I'm having trouble reading an MRI or something really strange, we can just go in and ask them and get, an opinion and go over the thing. So it is really good for quality of patient care. And there's 5 neuroradiologists. They're going to hire a couple more. And the people in radiation oncology, radiation is very effective, very commonly used therapy in our field. And there's 4 neuroradiation oncologists who basically just do this, including Dr. Ahmed, who'll be talking tonight. And then finally with neuropathology, that has to do with looking at the tumors themselves when they're on slides, say after somebody had surgery. And it's super important to have people who can interpret what the tumors exactly are, not in terms of their appearance and where they might have come from, in addition to that, but the molecular markers and are there any proteins that are expressed that might be important for treatment. So we spend a lot of time figuring out the tumors, what their basic characteristics are, and which proteins they might have for which there might be a target. So it's super important. And then I left someone off here. We do have a personalized medicine person, Dr. Walco, who actually can take a lot of the molecular information that we get and make treatment recommendations on that basis and tell us about clinical trials, say, that are outside our center. So that's sort of the 5-minute introduction. And so I just wanted to introduce Dr. Ahmed, who is now an associate member at the Department of Neuro-Oncology. He's really an outstanding physician and an outstanding researcher. So he received an award, I think, 2 years ago, Cameron, for the up and coming clinical researcher of the year, which means a lot. He does a lot of clinical trials and is very productive and a great colleague. So with that, Dr. Ahmed, I'll probably leave you alone and let you carry on.
Thank you so much, Peter. Appreciate that introduction. And as you mentioned, all of our patients we manage as a team. So I'm a radiation oncologist, and I have a clinical and research interest in the management of brain metastasis. And I'm just going to be going over a few slides in terms of the clinical management of brain metastasis. So brain metastases are the most common intracranial tumors in adults. They comprise about 200,000 new patients who are diagnosed with brain metastasis per year. And given brain metastases can arise from solid tumors, lung cancer, breast cancer, melanoma, and colorectal, they're up to 10 times more frequent than primary brain tumors. Ultimately, about 20 to 40% of patients with cancer may be diagnosed with brain metastasis, with the most common histologies being lung, breast, and melanoma. Next slide. So how do brain metastases develop? So, it's believed that the primary tumor cells, coming from the primary tumor in the lung or the breast or from melanoma cells, these cells will circulate within the systemic vasculature, and then ultimately these cells will arrest in the capillary beds that align the blood-brain barrier. And then slowly these cells can extravasate into the brain parenchyma itself and then coapt the vasculature of the brain and ultimately grow and develop within the central nervous system. Next slide. So most commonly, brain metastasis will occur within the gray-white matter junction due to a decrease in the diameter of these blood vessels. The majority of brain metastases are diagnosed in the upper portions of the brain, supratentorial cerebral hemispheres, just owing to the greater volume of a brain that is present there. And then about 15 to 20% are in the cerebellum, and then a smaller portion are within the brainstem, 5%. So the majority of brain metastasis, when they are diagnosed, are diagnosed when patients are asymptomatic, which is good because then there's quite a few ways that these patients can be managed. However, ultimately, what might lead to patients having a brain MRI scan might be due to new symptoms such as new headaches, altered mentation or cognitive function, focal weaknesses, or altered mental status, aphasia, ataxia, seizures, or visual disturbances. So about 30 to 40% of patients who are ultimately diagnosed with brain metastasis might have symptoms at the time of their diagnosis. So there's a few different prognostic models that are available. This is one that is available online, but it's known as the BrainMet GPA. And it's separated out into different primary cancer types. So if patients are diagnosed with lung brain metastasis, melanoma, breast cancer, renal cell, or gastrointestinal cancer, it gives us some idea generally of what the overall prognosis is for patients who are diagnosed with brain metastasis of this primary tumor type. So for each of these, primary tumor types, there are different factors that are looked at. So for breast cancer, different subtypes, for lung cancer, different mutations, looking at the number of brain metastasis and the performance status that patients have. But generally, this gives patients a score, which can correlate to overall prognosis for patients. So this is one of the models that's available online for patients to look at themselves. So, due to the blood-brain barrier, local treatments historically have really been the key to management, and today also still are really the foundations of management for brain metastasis. So, for those patients who have fewer lesions, if patients might be symptomatic from a brain metastasis, what can really help the most is sometimes surgically removing the brain metastasis um, um, to help with surrounding edema and symptoms that patients might be having from the metastasis. And this can be followed with radiation therapy, or sometimes patients get preoperative radiation therapy as well to prevent these cells from seeding. In addition to that, patients who have, more focal brain metastasis, we have data for up to about 15 lesions now being treated with focal stereotactic radiosurgery. That's certainly an option. But for patients who have more diffuse brain metastasis, patients who may have a poorer performance status, oftentimes what we use is whole brain radiation therapy. And this is also commonly what's delivered for patients who have leptomeningeal disease as well. Next slide. So this is generally what the differences look like in terms of the radiation plan for patients who receive whole brain radiation therapy versus stereotactic radiosurgery. So whole brain radiation therapy is traditionally delivered over 1 to 2 weeks, and it's low dose radiation therapy that's delivered to the full brain. For those patients that receive stereotactic radiosurgery, it's focal radiation therapy, and it's delivered over 1 to 5 days, and it's high dose and more focused towards the singular brain metastasis that patients have. So what are the side effects of brain radiation therapy? So in the short term, patients can have fatigue, scalp irritation, hair loss, nausea, vomiting, headaches, muffled hearing. Oftentimes, these short-term side effects can be managed quite well with over-the-counter medications. In terms of long-term side effects that we watch for, so one of the side effects that we've learned a lot more about from the long-term effects of whole brain radiation therapy is short-term memory loss. And there's a few things that we now do to help mitigate this risk. And then the other from the receipt of stereotactic radiosurgery is damage to normal tissue, which can be known as radiation necrosis. And there's different ways that we manage this as well too with medications, including anti-inflammatory medications like steroids. So one of the ways in which we have now learned to decrease the effect of short-term memory loss as a long-term side effect from whole brain radiation therapy is the delivery of hippocampal avoidance whole brain radiation therapy. So this decreases the amount of radiation dose that's being delivered to the hippocampus to a far lower dose than is being delivered to the rest of the whole brain. And so we've learned that long-term patients who receive this sort of radiation therapy technique using hippocampal avoidance have a lower risk of having short-term cognitive issues from the receipt of whole brain radiation therapy. We've also learned about the use of memantine for patients who are receiving whole brain radiation therapy. This can also decrease the risks of short-term cognitive issues from the receipt of radiation therapy. So in addition to local strategies like surgery, radiation therapy, we've learned a lot more about using systemic medications to tackle brain metastasis and leptomeningeal disease. So there's quite a few targets now that have been identified in different primary tumor types that have been shown to be beneficial in the management of brain metastasis. So for lung tumors, ALK and EGFR mutant tumors have been found to have certain targets that can be used to combat brain metastasis. For patients who have HER2-positive breast cancer, there's a number of different medications that are available. Also, immunotherapies have been shown to be very beneficial in melanoma as well as lung brain metastasis, as well as CDK4/6 inhibitors in breast cancer. So these are two important studies that have been published, over the past few years, which have really shown us the power of using systemic therapies for the management of brain metastasis. So these are images, from a patient who was treated with nivolumab and ipilimumab. Moffitt was really one of the leading accruing sites on this clinical trial, which was led by Dr. Forsyth here at Moffitt, where we looked at the receipt of nivolumab and ipilimumab for the management of melanoma brain metastasis. And you can see here the images of a patient who had brain metastasis, what it looked like at baseline, and then 1 year later, the remarkable response that this patient had. One of the important compounds that's been shown to be beneficial for HER2-positive breast cancer recently has been tucatinib. And so this is commonly employed now as well, too, in the management of HER2-positive breast cancer. One of the things that we're doing here at Moffitt and others are doing across the country is looking at combining systemic therapy with radiation therapy. So we've learned more about how we can sequence radiation therapy with systemic therapy and potentially use radiation therapy to open the blood-brain barrier and then deliver those medications in order to have a larger receipt of systemic medications into the blood-brain barrier. So these studies suggest that potentially the opening following radiation therapy is most consequential about a month after the radiation therapy is delivered. And so, it's important that we study this more. And on the next slide, we'll go into some studies that are looking into this at Moffitt. So, at Moffitt, we're all about research and really pushing the field forward. So one of the primary ways that we do this is by having clinical trials that are here, that are our own clinical trials and cooperative group trials that are open here at Moffitt that are really pushing the field forward in terms of management of brain metastasis and leptomeningeal disease. So these are a few of the trials that we have open here at Moffitt. We look forward to really pushing the field forward and using research, from the laboratory to really push this science forward. And Dr. Smalley is going to be talking about that. So thank you all so much.
I wanted to provide people with some vision of how we kind of make discoveries and how research helps us work towards curing these tumors, which of course is what Moffitt is all about and why we all show up to work. And Ina is a scientist who's also an award-winning scientist. She was the up-and-coming basic researcher of the year this year. So we had like 2 award-winning speakers tonight. And she's just really closely and passionately involved with trying to figure out how cancer gets into the brain and how we can do a better job at treating it. So with that, and she's a great colleague. So with that, I'll get out of your way and let you proceed.
Thank you, Peter. So one of the things that is really amazing about Moffitt and being able to do research on Moffitt as a scientist is the close clinical collaborations that we get to foster. And this is really the thing that drives forward new therapies, new opportunities clinically. So I'm going to talk a little bit about how we do this. One of the things that myself and other scientists like me working at Moffitt strive for is translating a lot of our basic science into things that actually impact patient care. And the way we do this is when patients agree to participate in research and they donate tissues or samples, we actually utilize these to both characterize the samples to find out, learn more about the biology of the disease. And we also use them to create new models. Because when we make discoveries, one of the important things before we can, take them to the clinic is to make sure that they're true, to validate them, to rigorously test them and make sure they're safe and they actually do what we think they do. So this is, the kind of the workflow that we follow. After we make some discoveries, we utilize some of our preclinical models to make sure that they work well, and then we work back with the clinicians again to kind of translate these discoveries into the clinic. So I actually got into the neuro-oncology space by when I first started collaborating with Dr. Forsyth about 12 years ago. I kind of had an interest in, I was working in melanoma, basic science. I was really interested in drug resistance. And one of the kind of, at the time, latest frontiers in that space was drug-resistant melanoma brain metastasis. And when I started working with Dr. Forsyth, one of the things he had mentioned to me is, brain metastases are great, but you should really learn something about leptomeningeal disease. He said, it's this devastating disease. We have nothing, we know nothing about the biology. We don't have very good therapies. Why don't you come to the clinic so I can show you what it's all about? So I did, and one day I met this beautiful young woman who was a leptomeningeal disease patient, and she kind of, told me about her experience, and she was very, very passionate about participating in research, and that really inspired me about the potential of a close collaboration between patients, clinicians, and researchers, and what we could achieve. And the fact that this disease was, we knew so little about it and had impacted patients in such a dramatic way, it really inspired me to go into this space. And actually, I ended up pretty much dedicating my career to studying leptomeningeal disease. So it's this kind of interaction that we have at Moffitt among patients, clinicians, and scientists is really transformative. Next slide. And some of the things that power what we do are, of course, these resources that are pretty much world unmatched that we have here, especially in the space of brain metastases and leptomeningeal disease. We have extensive tissue and blood banks. We actually have the biggest CSF bank in the world. We have extensive efforts and collections of clinical trial specimens. This is basically when patients go on to a trial. We also collect specimens, prior to the beginning and maybe during the duration of the trial. And that really helps us understand, why some patients do really well, maybe why some patients don't do as well, and what we can change to improve that. We have a lot of state-of-the-art instruments and facilities that help us with this. And most importantly, we have a fairly unique interdisciplinary team of experts. A lot of basic scientists tend to work fairly independently. They're really interested in their kind of unique niche thing. And, they go into their lab and they do their thing. But here, what we try to do is we try to work as a team that has people from all kinds of different backgrounds, bioengineering, basic biology, mathematical modeling, medicinal chemistry, drug discovery, immunology. And we all look at the same problem through many different lenses, many different perspectives. And we find that in doing so, we're actually able to learn much deeper about these diseases and find out better creative ways to not only make discoveries, but also how to apply them clinically. And of course, we couldn't do this without cutting-edge technologies. That we're very thankful for this kind of support that we have at the institution. We have instruments that allow us to study tumors at very fine single-cell resolution. This is important because it allows us to look at individual tumor cells, individual normal cells, how they interact with each other, what each one of them is up to. Not, we think of a tumor traditionally as a whole, but really different cells within it could be doing different things. And we really need to understand that to be able to treat it better. We also are empowered to study the tumor as a whole ecosystem. We have technologies that allow us to do that in 2 and 3 dimensions. We also have world-leading animal models, mouse models, predominantly for central nervous system diseases and cancers mostly. And we, of course, have world-leading imaging technologies, both for cells and for mice. Now, some of you might be wondering why we want to study the tumor as a whole. Really, you should think about this as an ecosystem. The tumor cells don't exist in isolation. And I think a good analogy for this is like a garden. Cancer is like a weed growing in a garden. You have a soil that provides nutrients and support. That could be growing within certain organs and having access to certain nutrients, certain growth factors that promote tumor to thrive in a certain space. You can have other cells that are kind of normal cells that are part of this environment that could be similar to other plants that you would have in a garden, and some of them can inhibit growth. They don't allow access to the sun. Maybe they compete for different nutrients, water, resources. Maybe they provide a physical barrier to prevent the weed from kind of spreading out. And then you also have the gardener. The gardener could be your immune system, or even different therapies that kind of take care of other plants, but also surveil for weeds and these tumor cells. You can have a gardener that maybe is exhausted and can't see these tumor cells or these weeds. And so this is, in order to understand what is happening in the garden, how to best allow it to flourish, we really need to understand each component. Some plants grow really well together. Some, like crops, we know that having one really helps the other. We also understand that about tumors. Sometimes having certain normal cells help tumors thrive. So we can't look at tumor cells in isolation. We really need to understand the context that they exist in and how other cells might be helping them, how other cells might be mediating their responses to therapy. We also have the state-of-the-art cancer model systems that kind of help us understand more about basic science of tumors. And this spans from things as simple as tumor cells growing in cell culture. We have isolated and established many primary tumor cell lines or primary tumor cells from patient specimens that we receive from the clinic. We have devised special media formulations that we grow them in that try to mimic as best as we can the physiological environment that this tumor would encounter in the patient. In the middle picture there, you see we've actually taken that a step further, and now instead of just growing these cultures in 2-dimension, we actually have these very intricate 3-dimensional cultures that live in this liquid-like solid matrix that then we can put the tumor cells in there. We can put other normal cells that are usually components of the ecosystem. We can put immune cells in there. In this example, the green cells are actually tumor cells, or I'm sorry, the green cells are normal stromal cells and the red cells are tumor cells. We can study how they interact. We can, in this instance, we were perfusing this culture with cerebrospinal fluid from the same patients as we got the tumor cells from. So really trying to recreate the full tumor microenvironment so we can capture the correct biology. And of course, we have some world-leading mouse models for central nervous system tumors, including both brain metastases and leptomeningeal disease, and including models that are immune competent, which means they have an intact immune system, so we can really study the full intact stromal environment as well as the immune environment. So right now we're also working on making a brain and leptomeninges on the chip technology that will allow us to study and test new therapies for these diseases. So I'm going to quickly highlight 3 projects that we've kind of developed on the basic science side with the help of collaborations with patients and clinicians. In this first project, we actually identified a vulnerability of central nervous system metastases. In this case, it was specifically leptomeningeal disease from melanoma. And in studying the tumor using these kind of cutting edge 3-dimensional technologies and 2-dimensional technologies, it allowed us to identify an interaction between tumor cells and normal brain stroma. These are your supporting cells that provide structure to tissue. And we found out they're actually helping tumors survive in the cerebrospinal fluid environment. Without those cells, actually, the tumor cells just die in that kind of environment. But we found their interactions help the tumor survive and help the tumor thrive. We actually identified molecularly what controls that interaction and found a way to stop it. So this is something we're thinking about how to translate clinically and best attack this mechanism. Next slide. This is a project where we're also looking at how tumor cells can hijack normal cell biologies to survive. So in this instance, we're looking at the metabolic cooperation between melanoma cells and astrocytes. Astrocytes are, again, kind of part of normal tissue physiology of the brain. They usually help neuron cells kind of survive stressful environments, recover from injury. And in this case, in brain metastases, we actually found that astrocytes help melanoma cells survive on therapies and thrive in nutrient-deprived environments. So we're looking at how this interaction mediates the survival and growth of tumor cells, also how it affects immune suppression in that space, and what are the mechanisms that basically turn this astrocyte from a normal kind of unreactive astrocyte into one that helps the tumor. And then in this project, we were looking at, we were trying to understand why leptomeningeal disease is so non-responsive to therapies, specifically immune therapies. And we also wanted to understand what's contributing to this kind of severe neurocognitive decline we see in patients. And what we found is actually there's, in the cerebrospinal fluid environment, in the leptomeninges, we see a big accumulation of branched-chain keto acids. This is just an abnormal metabolite that typically is not accumulated. This usually happens in inborn metabolic disorders. So this example of a child with maple syrup urine disease, in that instance, they do have big accumulation of these metabolites in their central nervous system. And if it's untreated, it's actually fatal. So what we found is that this accumulation actually promotes the suppression of immune cells. So they're basically inactive against the tumor. And it also promotes a neurodegradation. So we see death of normal neuronal cells. And when we applied a therapy that's usually used for these inborn metabolic disorders to our mouse models of leptomeningeal disease, we actually found that this therapy can be neuroprotective and help promote anti-tumor immunity, leading to longer overall survival. So this project really provided us a therapeutic opportunity for repurposing existing drugs that are already FDA approved for other diseases to this very severe terminal cancer complication. Next slide. So this last project is really the perfect example of what I introduced at the beginning of bench to bedside translation. So in that project, we started with clinical specimens from patients with leptomeningeal disease. We characterized them. We created new models. We made this discovery of this branched-chain keto acid accumulation in my lab, and then we tested these therapies in our preclinical mouse models, and now we're working with Dr. Forsyth and Dr. Pina in a neuro-oncology team to design a clinical trial in order to translate these findings into the clinic. And that's it for my part. Now I'll bring this back over to Dr. Forsyth.
Yeah, that was great. I mean, Dr. Smalley did all this metabolomic screening of the CSF that I was convinced wasn't going to lead anywhere. It's just a lot of work where there's, 100,000 different signals that you have to sort out what's important and what could be important. And it was surprising that it worked out so well at the end of the day to find a clinically available drug that was already approved and used for neurologic problems. I mean, it's just a great example of, I think, working hard, being creative, and kind of getting lucky. So we want to move that into a clinical trial as soon as we can. The other thing that she mentioned is that it looks like it protects nerve cells too. So we're interested in leptomeningeal disease. It's an uncommon complication of metastatic cancers. And to some extent, we just wanted to do something about this because there was almost nobody understands anything about it. So we're one of the top 3 programs in the country. There's very little that's understood about the biology, and we just don't like to see patients in clinic who have this problem. So we've made some discoveries that we're translating into clinical trials, but this is Sarah, who's a young woman who had melanoma, and she developed this sort of spread of tumor cells to the surface of her brain and down in her spinal cord. When I first saw her, she just had a few changes on her MRI, and it wasn't completely clear what was going on, but I told her she had leptomeningeal disease, and then she kind of wound up hating me for at least 3 months. And then she made a gift that we could study her body when she died, and this just shows what it is. The melanoma is the black tumor cells that are spread over the surface of her brain and down in her spinal cord. So it's a pretty serious problem. Next slide. So a few things that are necessary to make progress on this disease. One of the first things is to understand it better, which we'll talk about in a minute. The second thing is really just for us to make a commitment, never to surrender, really just to believe that we can cure it and stop messing around by just coming up with different treatments. So in terms of understanding, Dr. Smalley's made about 3 important observations that nobody else in the world has made. And there's Dr. Bohr at Sloan Kettering has also made an observation. It turns out that this fancy slide is just meant to show that there's a whole bunch of immune cells in leptomeningeal disease, which is surprising to me because I always thought in clinic that if you looked at somebody's spinal fluid, that there would just be a bunch of tumor cells in there, just hanging around and making people sick. But it turns out that that's a small number of the cells. So a lot of the cells are immune cells, which of course means that there's a strategy so we can manipulate the immune environment to come up with better treatments. And this paper that was just published in the most famous scientific journal in the world basically showed that in patients with leptomeningeal disease, they're kind of working hard on their own to try to fight the disease by making these different chemical cytokines and turning on some dendritic cells. And then without further treatment, we have things called natural killer cells, and they kind of attack the cancer, but don't really do a good enough job on their own. So we turned to this with these clues that dendritic cells might be important. We turned to manipulating the patient's own immune system and to educate it to recognize proteins that are present in their own tumor and then inject it directly into the brain. So can you do the next slide? So I had to start this in mice. So we take the mouse's or the patient's own immune cells, and we just put them in a dish for a couple of weeks with some of the proteins that are present on the surface of their tumor. And then we sprinkle it with this cocktail of different cytokines to turn on the cells and really kind of get them a little bit angry, but also to educate those cells to recognize and say, look, these proteins from this patient's tumor are bad. You need to report them to the rest of the immune system. This is a little too busy, but when we do this in mice and we put the dendritic cells directly into the spinal fluid, most of the mice, but not all the mice, are cured, at least in this HER2 model or HER2 mouse tumor. And this is a triple negative model where it works pretty well, but not as well in HER2. If you go to the next slide. And then this is maybe a little complicated, but it's a CD4 T cell therapy. So in this case, these are, I'm circling it, although you can't see my circle, but the yellow cell in the middle is a dendritic cell, which is getting educated so it can recruit immune cells to kill the tumor. And it turns out, surprisingly, that on the right-hand side, it looks like in mice anyway, that a lot of that occurs by turning on B cells, where these CD4 cells will turn on B cells. And B cells, of course, are the holy grail because they make antibodies, and antibodies to these proteins suggest it's possible to have long-term resistance. It's kind of like getting it's like getting a vaccine, right? For polio, say, I mean, once you've turned on your polio antibodies, you're never going to get polio. And that's true for most of the rest of the vaccines. Can you do the next slide? So this is I thought it'd have arrows, so this is just super colorful, but also pretty confusing. But we're doing a clinical trial, which we've treated 10 patients so far, and we've looked in detail at the individual cells in everybody's CSF after they've had treatment. So again, without the pointer, sorry about that, but you can characterize on the left, you can characterize the population of the cells, and which are cancer cells, for example, or which class of immune cells they are. So in the first patient on the top on the left, there's a whole bunch of tumor cells in the gray and the lilac colored thing. And all the other immune cells have increased as well after treatment. And that's kind of shown in the bar graph on the right, which you probably can't really read very much, but it shows that she has this tremendous increase in the number of immune cells. And somewhat paradoxically, unlike the other patients, she actually had more tumor cells. Now, she lived a long time and died about 15 or 14 months after starting the treatment because she had radiation damage to her brain, not because of the tumor cells. But it looked kind of like she developed a mechanism to live with the tumor cells, because when we started treatment, she was super sick. And then she was pretty normal, actually. And she allowed me to share some videos of her, with her family or with her girlfriends, where she was almost a normal person. And then this patient number 2 started out with some tumor cells, but they basically almost went away completely. And she had this big induction of all these other immune cells that you can't really tell what they are, but they're the green, purple, and orange cells. And a lot of these are immune cells that mediate memory, which of course you want a memory in your immune system. And a lot of them are B cells, the one that make the antibodies. So that's super encouraging. I've kind of seen this with everybody. And then at least in the first 6 patients, I mean, patients on average live 2 or 3 months. And so far on average, people have lived more than a year, which is a long time. So we have to do better, and we need to see how to make this better. But that's a different story. The next slide. And then just to test this, which is a long story, we actually do detect, you're going to have to trust me on this one because it's not obvious, but out of the 5 patients we tested, 4 of them had antibodies to the proteins that were of interest that are present in their tumor, HER2 and HER3. So it's pretty remarkable. To make a long story short, we sort of proved that we could turn on the immune system in a way that would be at least somewhat specific to the proteins that are in somebody's tumor and leptomeningeal disease. And I think most of the patients that had that happen are still doing well. It's not a perfect treatment, but it's super encouraging. Do the next slide, please. And so that's just an example of that's a Moffitt science thing that Dr. Zernicki's worked on dendritic cells. So we used his ideas and technology, collaborated with him. We have lab meetings together and grants together. And now our next project, this slide is meant to kind of summarize in way too much detail for today, but on the right-hand side, we want to take the uneducated parts of the immune system that I call kind of a bit of a machine gun, but you want to turn on the machine gun approach if the enemy's attacking you and they show up and they start storming your house or something. You basically just want to kind of defend yourself and get on with it. And then on the left-hand side, it's more an adaptive thing or an intelligent or selective thing. So while you're defending yourself in the next few minutes, you want to be assembling some kind of specific thing where you're just going to kill the enemy with a single bullet. I'm sure the gun thing is not the most appropriate analogy, but you have kind of a sniper rifle approach. So you're just killing the enemy tumor cells and not everything. So we're going to take this early success we have and build on that by adding other therapies like the Herceptin that Dr. Ahmed and I are doing a trial in now, because those things on their own, they both work pretty impressively. So can we combine them and see if doing them together works? Or for triple negative patients, we want to give intrathecal other immune therapies like nivolumab, for example. And as Ina said, behind all this, we do studies to study people's tissue and specifically to see if it has the effect that we think it does, and if it's working in someone or not working in someone, and should we change therapy. So that we heard that we're funded, we don't have official notice, but that's to do a couple clinical trials. So that's really exciting. Can you do the next slide? So that's an example of the immunocellular therapy approach that Moffitt's famous for. And we think that we can, we're going to work towards curing this disease, and we can do that. We're the third biggest cancer center in the country, and we all know that America is the country that is going to cure cancer, so why not do it to LMD? This final slide is a little more controversial, but I'm aware we need to improve the patient caregiver experience. So we'd like to work, which we can't do right now, but we want to work in the future so we can really have all the care revolve around the patient. So our optimal situation that we'd like to develop is to have a patient come for kind of one visit and see radiation and medical oncology, neuro-oncology, and neurosurgery in one visit. So in this case, it's like having the patient's the sun around which the planets all orbit, rather than the way we're doing things now. It's kind of see one specialty one day and then say, oh boy, you have to come back tomorrow or the next week to see somebody else. So, or where we need to do a better job of that and really want to do that. And then it's supported by all these things on the outer ring, tumor banks, animal models, all the chemistry and immunology that we can do to do profiling, as well as have some bioinformatics to look at who's being treated how and by what and how are they doing. So I think with that, I'll stop and say that's our vision for to cure these tumors and develop better treatments for them. And Tyler Wilson, you represent the foundation. Were you going to say a few words?
Yeah, and just thank you so much for the opportunity to be here tonight and be part of this presentation. We're grateful for the opportunity to partner with the Neuro-Oncology Department. And really, I think tonight's presentation was a wonderful example of the impact that philanthropy can have really across the cycle of research, whether that's with Dr. Smalley at the basic and translational level, or some of the presentations from Dr. Forsyth and Dr. Ahmed at the clinical level, and seeing research evolve from a petri dish into a clinical trial and then move into a potentially a partnership with the DoD on the grant that Dr. Forsyth presented. So at every step of the way, philanthropy can really make an impact, and it's the cumulative impact of every gift that we receive here at Moffitt that can really have that impact. So I've been with Moffitt now for 5 years and been partnering with the neuro-oncology team for a majority of that time. And it's really been remarkable to see some of the advances that they've made possible and the impact that philanthropy has had. And philanthropy is definitely a personal decision. And so if and when you are ready to make a gift, my contact information is on the screen. I'd be more than happy to help you go through that process and share with you, learn more about the impact about making a gift. And then, at Moffitt, we try to make it as simple as possible to make a gift. And so if you would like to make a gift and designate it for neuro-oncology research, you can simply note that online or on the memo line of the check. So again, thank you so much for the opportunity to be here tonight and be part of this presentation. And now we can transition back to Dr. Forsyth for the Q&A.
Next year, we'll talk about some of how we manage the neurologic complications and try to get better at doing that. But it's definitely a huge problem. So I think Cameron, can you see the chat?
Yeah, so I think the question is regarding RILP, which is radiation-induced lumbar plexopathy.
Oh.
So this can happen, as an unfortunate side effect for patients who receive high doses of radiation therapy to the lumbar plexus. So there aren't, I think, great, it's an unfortunate kind of side effect. The main things are kind of managing the side effects of RILP. And that can include, medications for the neuropathic pain. There's other medications that aren't necessarily, for neuropathic pain, but, other ways of kind of preventing, this might be, I think, vitamin E pentoxyphylline was mentioned. And then sometimes there's, I think, a little bit of data about hyperbaric oxygen as well, too.
Oh, yeah, that's a good idea. What about, does Boswellia do anything?
So Boswellia has been studied for radiation necrosis. It hasn't been studied as much for lumbar plexopathy or brachial plexopathy, but that's an interesting thought. I think it's the same mechanism that's in play, so might be worth studying that.
I mean, the problem with the nervous system is that it's just very vulnerable and difficult to repair when it's been damaged. So there's been, decades of research into spinal cord damage, peripheral nerve damage, looking at nerve growth factors and all sorts of other things, but it hasn't really paid off yet, actually.
Mm-hmm. We do need better management strategies for this.
And some kind of customization, because you'd like to be able to tailor like how much is enough treatment for this patient, but it's too much for somebody else, and we really need to move that way, but we don't really have all the tools.
Mm-hmm.
We run into this problem a lot with other immunotherapies affecting the brain or spine or nerves all the time, and we're going to talk about that next year, I think. So maybe we should move on. Oh, Cameron, here's another one for you. Can you see the second one about what are protons? And
yeah, so protons are a different type of radiation therapy. So most commonly, photons are what we administer for radiation therapy. There's a lot of interest in using protons to spare normal tissues, from the side effects of radiation therapy. And one of the important areas that it's being studied right now is in the management of leptomeningeal disease when we're using craniospinal irradiation. Because it can help decrease the amount of radiation dose that's going to the bowel and to the bone marrow as well, too. And so there are other strategies that we kind of use to mitigate radiation dose to those normal organs when we're not using protons, but Moffitt is going to be getting protons as well, too. I think the center is going to be opening in Spiros in 2026, so we're very excited about that.
Yeah.
And to offer that to our patients.
Just wanted to remind people, if we don't get to all are we only going till 7 o'clock, is that right? Oh, so if we don't get to all your questions, we will follow up on Zoom.
Brielle said we can go a few minutes over as well.
Did we vote on that one?
Yeah.
Okay, who's going to do, next is Next is how many times can you have stereotactic radiation to the brain, and how do you prevent necrosis?
Yeah, so that's a
Dr.
good
Ahmed, can you do that?
That's a good question. So there isn't really a limit in terms of the number of times that patients can receive stereotactic radiation therapy to the brain. Um, the one thing that we are very careful about when we're retreating the brain is that we're not overlapping areas that have received previous radiation treatment. So whenever we have to retreat an area with stereotactic radiation therapy, we're more cautious about that because we want to make sure that we're actually treating cancer at that point and we're not treating, some of the dead necrotic tissue, which can then lead to more side effects. So not really a limit in terms of the number of sessions of stereotactic radiosurgery to the brain, more so just making sure we're not retreating an area. And then how do we prevent necrosis? So another good question. And so there are, medications that we commonly will use to help prevent that. So one of the ones we were talking about earlier, Boswellia, that's something that we'll use to treat vitamin E pentoxyphylline. We also use Avastin. And then if the side effects are more consequential, we also use steroids a bit as well too.
Can I just ask a follow-up? Is there anything specific with younger patients? So I think that patient's a younger patient.
So not, yeah, I mean, we know that the risk is probably increased over time. So, obviously we want to decrease the amount of radiation dose that we as much as we can for younger patients because we know that there's a long life ahead of them. But in terms of kind of specific strategies, in terms of medications, that's still kind of, I think we need to do more research there.
So, you want to do the next one about AI?
Yeah, sure. I mean, we are actually currently employing AI in research. So it's something at the forefront. At Moffitt, we're employing different machine learning and AI techniques to enhance how we analyze imaging, how we interpret data, how we model tumor microenvironment. So it's definitely something that is very much involved in the research side right now. We actually have a whole machine learning department that's got established, maybe 2 years ago now, that is thriving at the moment, recruiting faculty. So I'm expecting that you'll see a lot more about that in the coming years.
I think the key is getting really good data, and then it's so complicated that AI is going to help our tiny brains
Yeah.
sort out relationships that weren't apparent, so
Yeah.
I know it's pretty interesting.
Mm-hmm.
Um,
And some of the questions we're also answering typed, and they're just showing up on the answered tab in case you asked a question that's already been answered.
what was that? Were you telling me something?
No, just for the audience, because I know some people ask questions and I'm trying to type answers for some of them.
Yeah, I did some too, yeah.
And so they're disappearing and reappearing under
Yeah.
the answer tab.
And then the next testicular LMD, geez, my boyfriend's 27 years old, got diagnosed with AMD, being treated at UCSD. But we're worried about proton therapy because they say don't really understand his situation. Yeah, because I'm not sure how to answer your question. I mean, I haven't seen a patient with testicular cancer who had leptomeningeal disease either. I've really only been focusing on it for about the past 10 years. Cameron, do you know just about protons? And because they're it's so sensitive usually, right? Would there be in terms of the testicular cancer responding to chemotherapy or radiation, do you know anything about testicular cancer?
So I would say that it is rare, to see testicular cancer metastasize to the leptomeninges. In terms of proton therapy for the management of, leptomeningeal disease, we have seen good outcomes treating the craniospinal axis with proton therapy for leptomeningeal disease, in a phase 2 trial that's now being studied in a phase 3 study. So I do think that, particularly for patients where that's the sole site of their metastatic disease, that, it is, a good treatment to be offered.
So what's next? The acceleration resulting of LMDM27 is wondering if I do a trial. Sure, I can answer that. So maybe, Ina, you want to do the maple syrup urine disease question? Can you see that one?
Yeah, so to talk more about the medicine to treat maple syrup urine disease is currently available for LMD. So the therapy is actually sodium phenylbutyrate. It's also available as glycerol phenylbutyrate for maple syrup urine disease patients. It's FDA approved that it's fairly safe. We are actually in the process of writing up a clinical trial to test this and hopefully get some funding to actually support it. It's a little bit challenging because this compound is not currently under any IP, so there are no companies that would be interested in funding this. So we need to find a way to kind of push this into the clinic. I don't know, from a compassionate care, is that something that's available to patients, Peter?
Well, the problem is you'd have to fund it, so we just so everybody knows, we don't walk around with our hands out. It's just that we have to apply for peer-reviewed funding through the NIH and NCI, which is a bit complicated now. And then even for the best of the best applications, maybe 5 or 10% of the applications are funded. So we're going to apply for that. Virtually all the work we do is already peer-reviewed funded, if you know what I mean, but it definitely would benefit from a leg up in terms of philanthropy. We're not looking for something for free. We just want a hand to get it into our patients and help, I don't know what time was it, so let's see. I was going to answer this one about the breast cancer. Tumors in another part of the body affect a patient's mental condition. Yes. It can. Tumors elsewhere in the body can do that to you for several reasons. One is that, first of all, it's very distressing having cancer. It's a full-time job looking after you or yourself or a loved one once you have cancer. And that in and of itself can really affect somebody being rude or combative, as you say. A lot of people are on steroids, and steroids can really make people kind of crazy, very aggressive, eat a lot, can't sleep. Occasionally it's very, very severe. So some of the medications can also affect somebody's mood in addition to steroids. And then there's rare tumor syndromes where our immune system kind of attacks the tumor cells, but it also can attack some of the nerve cells of the tumor and the nerve cells. So they're share the same antigen. So paraneoplastic syndromes is a rare, super rare cause of some of this bizarre, crazy behavior. We're just doing this in, fasting in the brain tumors? I have no idea. Cameron or Ina, do you have thoughts about fasting? Is that certainly not going to be sufficient? Having a healthy diet is good. Not starving yourself. Don't starve yourself. That's just bad.
Yeah, not sure. It's even with our keto acid story, because keto acids come from amino acids. So for maple syrup urine disease, one of the treatments is to restrict amino acids in their diet, specifically branched chain amino acids. But this might not work for cancer patients because they have other complications like cachexia, muscle breakdown, that might eliminate any benefit from that kind of diet change. So it's something we're investigating, but not sure about an answer for that.
So keto acid or anti-inflammatory, I don't know that that helps.
Yeah, again, the keto diet, I don't think that would restrict, at least in relation to the keto acid story, it wouldn't really restrict amino acid availability. But again, not too sure about the rest.
Yeah, and I don't know about this last question about immunotherapy. I mean, it depends what kind of immunotherapy you're thinking about. I think that none of the three of us on the call know enough about testicular cancer because it's a very specific thing. You know, we could set up a Zoom or something or talk about it, but I don't know that immunotherapy has been useful in testicular cancer. I'm just ignorant. And again, since I don't think I've seen a case of leptomeningeal disease from testicular cancer, although I'm sure it happens, I really don't know. So maybe we've reached, I think, the end of, we answered all the questions, Brielle? Okay. So look, we're very, very grateful for everybody to listen and ask your questions and participate. We're pretty excited about what we do, and just wanted to thank Dr. Smalley and Dr. Ahmed for being great colleagues and helping out so much and helping out tonight. So with that, we'll say good evening, if that's okay. All right. Thank you. Thank you very much.